Hello, I'm Ashley Dawson, a research resident studying aortic disease under Dr. Zhang Shen and Scott Lemaire. Thank you for the opportunity to present our work. We aim to determine the cell-specific expression of critical ECM genes in the aortic wall in patients with Marfan syndrome. Samples taken from the ascending aorta of 3 patients with Marfan syndrome and 4 age-matched controls were digested to single cells and the data combined. As the genes of interest in the study were predominantly expressed in the non-immune cells, these were identified, extracted, and recombined for further analysis. General groups were identified using conserved gene expression, including that of smooth muscle cells, endothelial cells, mesenchymal stem cells, and fibroblasts. We identified clusters within each main group that exhibited changes in gene expression consistent with a modulated phenotype. Modulated smooth muscle cells, or smooth muscle cells with increased expression of extracellular matrix genes, activated fibroblasts with increased proliferation, angiogenesis, and smooth muscle cell markers, and dedifferentiated endothelial cells with increased contractile gene expression and increased expression of collagen genes to a level consistent with that seen in smooth muscle cells. The highest expression of fibrillin-1 was in fibroblasts and modulated smooth muscle cells. Expression of the gene encoding fibronectin-1 was highest in cells with modulated phenotypes, including activated fibroblasts, modulated smooth muscle cells, and dedifferentiated endothelial cells. Genes encoding collagen 1 and 3 were predominantly expressed in fibroblasts. Elastin gene expression was highest in dedifferentiated endothelial cells. Expression of the gene encoding lysyl oxidase was highest in fibroblasts, and the expression of genes involved in matrix turnover were also highest in fibroblasts, predominantly in quiescent fibroblasts. On analysis of differentially expressed genes, fibrillin-1 was found to be downregulated in Marfan syndrome in most fibrillin-1 expressing clusters. In Marfan syndrome aortas, activated fibroblasts in particular had consistent downregulation of genes associated with microfibril formation, including fibrillin-1, fibronectin, and LOX. In contrast, genes encoding collagen 1 and 3 and elastin were upregulated in fibroblasts and modulated smooth muscle cells in Marfan syndrome. In conclusion, critical extracellular matrix genes in the aortic wall were most highly expressed in the fibroblasts and modulated non-immune cells. Genes encoding collagen 1 and 3 were upregulated in Marfan syndrome. And although elastin genes were also upregulated, other microfibril genes were downregulated in Marfan syndrome, particularly in the activated fibroblasts. This may indicate a dysfunctional response to aortic stress. Thank you.